of things. Mutations. What are mutations? Basically, a mutation is an error in base sequence that is reproduced during DNA replication. What causes a mutation? Usually a mutagen. Some substance or agent that causes a change in the structure of the gene. It could just be a one nucleotide mess up. Those are called point mutations. There could be a stretch, a deletion of a few nucleotides. There could be something called a translocation where a whole section is moved over to someplace else. Many things could be mutagens. Ultraviolet radiation, cigarette smoke, there's, certain chemicals can cause mutations. And then there are these just random mutations that occur when DNA is replicated. The good news is DNA polymerase, remember DNA polymerase, that enzyme that's involved in DNA replication, it has some proofreading capabilities. So it kind of makes sure that the right nucleotides are complementary base pairing. But every now and then a mutation will occur and there will be some problems. A lot of diseases are caused through some type of mutation that happened a long time ago. If you think of the disease sickle cell anemia, there is just one difference in one nucleotide that causes sickle cell anemia, because what happens is a nucleotide in a codon that codes for the amino acid glutamic acid in normal hemoglobin is replaced with a nucleotide that switches over, and then the amino acid that's coded for is valine. Well, valine is a non-polar side chain amino acid, and now it's there instead of glutamic acid, which was polar acidic. Think about the possible bonding that glutamic acid would do, the ionic bonding in three-dimensional structures, versus now you have a valine that is hydrophobic, so the protein folds differently, and it causes all sorts of issues, all right? So you can read up about mutations and mutagens and all that stuff. Let's move on to the last part of this section, which deals with DNA recombinant technology, which back in the olden days, they used to call this cloning. DNA recombinant technology is actually combining two different DNAs from different organisms. So let's say you have a gene that you want to make a lot of, insulin, for example, or human growth hormone, or tissue plasminogen activator. These are all proteins that we use DNA recombinant technology to make a lot of them, all right? Make a huge number of them. What we have used, and what is used, for insulin, for example, is something called a plasmid. A plasmid is this piece of circular DNA in bacteria. So we got this bacteria, it's got a piece of circular DNA in it, and it's got some chromosomal DNA. All right, so you take out the plasmid from the bacteria, and then you cut it. And how do you cut it? I remember once when I was teaching this, a student in my class said, do we have teeny tiny scissors that cut it? No, we use things called restriction enzymes to cut the plasmid. So restriction enzymes. And these restriction enzymes cut the plasmid in specific known locations. So now we have a cut plasmid, and now we're going to insert, and this plasmid is DNA. Remember, we're going to insert this foreign DNA in red, okay? Insert the foreign DNA, and then seal it, ligate it, and then stick it back into the bacteria. So this goes back into the bacteria, and let's say here it is. Here we go. And bacteria will do what they do best. They will start to replicate. And now the two new bacteria have that recombined DNA in it. Okay? And then these will more, humans takes 9 months to reproduce, bacteria take much, much less time. So within a day, you got a whole bunch of bacteria that have all these recombined plasmids. All right. And then what you can do is trigger transcription translation, and then you get all the protein that you want after you translate this, and you got tons of insulin. So you don't have to depend on an animal's pancreas to get the insulin to give to all the diabetics of the world. You can actually synthesize it using plasmids. Okay, there are other vectors besides plasmids, but right now that's the only one you need to worry about. And I think that does it for the DNA recombinant technology. You can read the details of all the steps in your book, but we're all set, and I will see you later.